Okay, for our last debate of this session, we are going with CAR NK cells plus anti-CD20. Ioannis Parodis, who is a rheumatologist at the Karolinska, will lead off the debate.
Yes, thanks for inviting me. I'm happy to be here. And I'm not an expert in the field, and I don't even have any data. So I don't know why Brad invited me to talk about CAR NK cells, but I will do my best to put the scene and trigger discussions during the 3 days we will be here. These are my disclosures. So now the scene has been set for the auto CAR-Ts and for the allo CAR-Ts. And now we will be talking a little bit about the T-cell engagers or the cell engagers, the B cell killers, T-cell engagers, because we start having some data there in autoimmunity, but we will also talk about the CAR NK cells. And the big advantage that we would like to mention already to start with the CAR NK cells is their safety profile. So from the very limited experience that we start seeing with the use of CAR NK cells from allogeneic sources, we don't see any cytokine release syndrome and we do not see any neurotoxicity. So this is probably the big advantage of CAR NK cell therapy. So my colleague will have to defend the high tocilizumab use with the T-cell engagers and the high toxicity that we see at least in the hematological malignancies. So to start with the comparison between CAR T cells and CAR NK cells, why are the CAR NK cells better? To begin with, the majority of what we have seen so far from CAR T cells is CAR T cells from autologous cells. And what actually is engineered for CAR NK is from allogeneic sources. And this, of course, minimizes the risk for toxicity. And the big advantage of the NK cells as a cell, not with or without the CAR engineering, is the dual mechanism that these cells provide with the antibody-dependent cellular cytotoxicity. So this, of course, makes the combination of a monoclonal antibody with a CAR-engineered NK cell very appealing, and this is what I'm here to defend. Now, coming from allogeneic sources, the cost is lower compared to with the CAR NK cells coming from the individual that we will be treating. And of course, the off-the-shelf manufacturing also contributes to scalability. Now, comparing the monoclonal antibodies with the CAR NK cells, we see to the left, the monoclonal antibodies have the advantages that they are off the shelf, they are manageable, we have experience with them, and they are specific, they are safe. And to the disadvantages, they have the short half-life, and they require multiple administration, which maybe we will not have to the same extent with the CAR T-cell therapy. But this comes with the cost of toxicity, coming not only from the CAR T-cells, but also from the preconditioning that is required for giving this therapy. And this is also something that might be one of the big advantages with the CAR NK cells, because preconditioning here might not be required. And this is something that now is tested by, for example, Fate Therapeutics. CAR NK cells with and without preconditioning. So what the CAR NK cells aim to do is to combine the best of these two worlds. Now, the comparison between the CAR NK and the bispecific T-cell engagers becomes a little bit more tricky, especially because I actually believe in the bispecific, so I don't know how to how to debate against them. Okay, but to start with, both of them have the big advantage of the dual mechanism, especially with the CAR-NK combined with the anti-CD20. You have the dual mechanism with the CAR-NK, and this you also have with the bispecifics. And the bispecifics have the advantage that they are more mature in their development, especially for autoimmunity. And when it comes to the toxicity profile, here I believe we have the big advantage with the CAR-NK cells, because here we can expect a much safer profile. Again, the data, or the majority of the data that we have for the bispecifics, come from the hematological malignancies. This is where we see high rates of cytokine release syndrome, and we will see what the safety profile of the bispecifics is in autoimmunity. Now, when it comes to the immune escape, there might be an advantage here for the CAR-NK cells, especially since, apart from the cytokine release syndrome and the neurotoxicity, we don't have evidence of immunogenicity either. Some very early in vitro feasibility data that I have got the allowance to share with you, coming from the glycostem CAR-NK cell. Here we see that with different doses of the CAR-NK, we see a dose-dependent cytotoxicity. Here to the top, against healthy B cells, or B cells coming from healthy individuals. And then you see the combination with obinutuzumab that indeed results in enhanced cytotoxicity. So obinutuzumab alone, of course, depletes B cells. CAR-NK cells alone deplete B cells. Those together show a synergy. And what happens against B cells coming from SLE patients? Here we see 2 patients with SLE and CAR-NK cells coming from 3 different donors. And despite the donor variability, we see again a dose-dependent extent of the cytotoxicity that is induced from the CAR-NK cells, up to 90%. And again, combined with rituximab, we see that there is enhanced cytotoxicity when rituximab is given along with the CAR-NK. In this case, you see that the rituximab was given at the highest subtoxic dose of rituximab. So there is some kind of synergy, and what point I would just very briefly would like to bring up here is how we monitor the killing of the B cells. It is important to somehow use the right target, because to me, using the cell markers that we target is a little bit problematic for the monitoring. So here, using an untargeted surrogate, if you wish, B cell marker, the CD22, we again see the cytotoxic effect of the CAR-NK, both alone and combined with rituximab. Here, of course, the effect is lower, but it is, we can see 50%, up to 50% of cytotoxicity when the CAR-NK cell is combined with rituximab, but pretty adequate cytotoxicity also with the CAR-NK alone. So to summarize, the big advantage of CAR-NK cell therapy is the safety profile, the excellent safety profile. We start having some preclinical data for its use in autoimmunity. Of course, its use in hematological malignancies was what encouraged B cell malignancies, was what encouraged its use in lupus and other autoimmune or immune-mediated diseases. So we will see more of the CAR-NKs. The cost is lower, and of course, the advantage of the dual targeting. And what I also believe will be something that we will see in the tissues when we start doing tissue investigations is that the natural homing of the NK cells into lymphoid organs will result in better targeting of the tissue-resident autoreactive B cells, and this will also be one of the advantages. Again, the reliable monitoring has to take into account the reliable markers that we use for this monitoring. I would like to give a thank to the people that allowed me to share some of the data, and yes, time for questions we don't have, but we will be around.